Translation. The newly synthesized messenger RNA contains information in the form of codons. A codon is a sequence of 3 DNA or RNA nucleotides that correspond with a specific amino acid. Since these codons, 3 nucleotides, they are called triplet codons. Process of translation. After the messenger RNA is released into the cytoplasm, it reaches the ribosome where the process of translation occurs. Each messenger RNA can be made up of thousands of triplet codons. 2 other types of RNAs, that is, transfer RNA and ribosome RNA, also participate in this process. Each transfer RNA has an anticodon, which has the sequence complementary to the codon on messenger RNA. These transfer RNAs supply the amino acids as per the codons on the messenger RNA. The amino acids which are brought by the transfer RNA are joined together by peptide bonds. This process is carried out with the help of ribosome RNA. During this process, that is translocation, the ribosomes move from one end of the messenger RNA to the other end. At the end of this process, a protein is formed which undergoes further modification to form fully functional protein called complex proteins. These proteins control various functions in the body. This is how a complex protein looks. There are 3 main types of RNA. Number 1, messenger RNA. It encodes amino acid sequence of a polypeptide. Number 2, transfer RNA. It brings amino acids to ribosomes during translation. Number 3, ribosomal RNA with ribosomal proteins makes up the ribosome, the organelles that translate the messenger RNA. Structure of DNA. In 1953, Watson and Crick proposed a model of DNA molecule. According to this model, 2 parallel threads of nucleotides are coiled around each other. This arrangement is called double helix. Each strand of DNA molecule is made up of many small molecules called nucleotides. Each nucleotide consists of a molecule of nitrogenous base and a phosphoric acid joined to a molecule of deoxyribose sugar. There are 2 groups of nitrogen bases: A, purines, adenine and guanine; B, pyrimidines, cytosine and thymine. In a molecule of DNA, nucleotides are arranged like a chain. The 2 strands of DNA are comparable to the rails of a ladder. Each rail is made up of alternately joined molecules of sugar and phosphoric acid. Each rung of the ladder is a pair of nitrogenous bases joined by hydrogen bonds. Adenine always pairs with thymine and cytosine always pairs with guanine. Thank you, dear boys, for listening to this video. We are done with the chapter, and as I always said, reading will help in understanding what we have discussed.